Hello and welcome everyone, my name is Dr. Graham Cottrell and welcome to the first video on glycogen metabolism and glycolysis. So in this first video we're going to understand what carbohydrates actually are and how the body uses them, and importantly how the body then breaks down these carbohydrates, which are stored as glycogen, to provide energy. So what is a carbohydrate made of? Well, as the name suggests, it's made of carbon, and it's hydrated carbon, so we have hydrogen and oxygen. And carbohydrates are one of the four major classes of biomolecules. And here's a quick question for you: can you name the others? And of course, the others are proteins, lipids, and nucleic acids. Lipids you'll probably cover later in the course. Nucleic acids you should already know about, and we'll cover a little bit more in depth about proteins again later in this course. So carbohydrates can quite often be referred to as saccharides or polysaccharides or sugars, depending on the context within the textbook. Examples of monosaccharides include galactose, glucose, and fructose here in the image. And you see the functional groups in red, which are just slightly different. They're all 6-carbon sugars. We also have disaccharides, which are 2 monosaccharides together, and examples here are lactose and sucrose. And lactose is made of glucose and galactose, whereas sucrose is made from glucose and fructose. And of course you'll all remember lactose from the enzyme kinetics practical. So organisms need to store carbohydrates for many different reasons. One is the storage of energy. Another is for a structural component, so this is particularly cellulose in plants. But carbohydrates are also used as coenzymes, so these are non-protein compounds which are required for catalysis, and of course they're required for DNA replication. Energy is stored so that the organism can produce ATP on demand. An example of a non-protein compound required for catalysis is NADH, and you'll see this later in the module where we talk about ATP production. And of course, sugars are components of DNA but also of RNA, so therefore they're required for protein synthesis, making mRNA and proliferation of cells when the cell needs to copy its genome. So different organisms store carbohydrates in different ways, and animals store it as glycogen, and this is a multi-branched polysaccharide composed of glucose subunits, whereas in plants it's called starch. And again, this is a multi-branched polysaccharide of glucose, but this differs from glycogen mainly because the glycogen is more branched than starch, and that will become a little bit clearer what I mean by branched later in this video. So here is a sort of overview of the structure of glycogen. You can see here we have in a linkage where we have the linear linkage, we have the alpha 1 to 4, and of course this 1,4 refers to the number of the carbon atoms. When we have a branch, and these link the chains of glucose together, this is referred to as an alpha 1,6. Again, this is because of the carbon positions. And the glucose molecule in blue is referred to as the start of the chain, whereas those in red are what we call the non-reducing ends. And the importance of the non-reducing ends will become clear when we talk about the breakdown of glycogen. So glycogen is stored in the body in 2 main areas. This can be in the liver and also in skeletal muscle. Most of the glycogen is stored in in muscle in total, but it's very, very concentrated in your liver. So sort of per size, there is lots more glycogen stores in your liver. And glycogen is, pretty dense and can actually be seen by electron microscopy. And you can see here in the little black dots, and these are what we refer to as glycogen granules, and these contain all the enzymes which are required to either synthesize or to break down the glycogen. So why is it important that we understand how glycogen metabolism is controlled? Well, the synthesis and degradation pathways are important because they regulate the blood glucose levels. So this balance between synthesis and degradation must be tightly controlled. Synthesis and degradation are regulated by distinct pathways, and again this is an underlying principle of biochemistry, so that synthesis and degradation are regulated by different pathways, and you'll see this later in the third video when we start to talk about the breakdown of glucose in the pathway called glycolysis. So synthesis and degradation are always distinct pathways. And these pathways show the importance of enzymatic control, because all these pathways are controlled by enzymes. And there are a number of inherited defects in these enzymes which will regulate glucose metabolism, and you'll learn more about this in the Inborn Errors of Metabolism lecture. So how is glycogen broken down? So one of the enzymes that regulates this is called glycogen phosphorylase. This performs a phosphorylysis reaction, and what this does is it cleaves off one molecule of glucose 1-phosphate from the non-reducing end of glycogen. This leaves the main macromolecule of glycogen of N-1 or one molecule of glucose has been cleaved off to generate glucose 1-phosphate. But we also need to break down the alpha-1 6 linkages, and this is done by a different enzyme called a debranching enzyme. So the glycogen phosphorylase cannot do this, and this is performed by another enzyme. And this has two distinct activities, a transferase activity and a debranching activity. And these have slightly different names in glucanotransferase and alpha-1 6 glucosidase. And the action of this enzyme leads to the release of a single glucose molecule. So the phosphorylase will generate a glucose 1-phosphate, whereas the debranching enzyme leads to the release of a single glucose molecule. So how does this actually work? So if we look at a molecule of glycogen, you can see from the ends, with the non-reducing ends, glycogen phosphorylase will cleave those glucose 1-phosphate molecules from the glycogen. However, we then reach a point where there are 3 glucose molecules left, and this is where the transferase activity of the debranching enzyme comes in. And it transfers those 3 molecules onto the other chain. This then allows the glucosidase to then cleave off the remaining glucose molecule, resulting then in another chain which can then be cleaved by the glycogen phosphorylase. So I have a quick multiple choice question for you now. So which one of the following statements about glycogen is incorrect? We have A, breakdown yields glucose 1-phosphate and glucose. B, glycogen is broken down by an enzyme with 2 activities. C, glycogen is broken down by glycogen phosphorylase. D, glycogen is stored in muscle and liver. Or E, the branches are formed by alpha-1 4-glucose links. Remember, I'm asking which one of these statements is incorrect. And if you would like to have a go at the question, I'll give you a second just to pause the video before I reveal the answer. Okay, and the answer of course is E. The branches are formed by 1,4,6 links. This is incorrect. The branches are actually formed by the alpha 1,6 glucose links. All the rest of the statements there are correct. So we've seen that the debranching enzyme and the glucose phosphorylase generate two distinct products, the glucose 1-phosphate and glucose itself. So what happens to these products? Well, glucose 1-phosphate is transformed by an enzyme called phosphoglucomutase. So this moves the phosphate from the 1 position to the 6th position to generate glucose 6-phosphate. Glucose 6-phosphate can then move either into glycolysis, which we will discuss later in this series of lectures, or glucose 6-phosphate can be converted to glucose by the action of glucose 6-phosphatase, or glucose itself can then be fed back into glycolysis by the action of hexokinase. And again, hexokinase is an enzyme that you will have covered a little bit already in the enzyme lectures, but we will cover in a little bit more detail in the part 3 of this lecture series. Different tissues use the breakdown products differently. So two of the major sites are the skeletal muscle and the liver, which we will consider. So in skeletal muscle, the breakdown of glycogen, glucose 6-phosphate will always enter glycolysis. Whereas in the liver, glucose 6-phosphate will be cleaved by the glucose phosphatase, releasing that phosphate, converting it to glucose, and then the liver then releases this glucose into the bloodstream, which can then serve as fuel for the brain. And it's important to note that skeletal muscle and the brain lack the enzyme glucose 6-phosphatase, and this is why the glucose 6-phosphate in skeletal muscle and brain cannot be converted back to glucose. The liver is the only site, or at least one of the only sites, that the glucose 6-phosphatase exists. And if you'd like to do any more reading about glycogen metabolism, then I recommend chapters 15 to 19 in Straya. And thank you very much for listening.